Today, we're going to be molecular detectives. We're going to unravel a story of how something incredibly small, a tiny typo in our DNA, just 2 letters long, can have an absolutely massive and sometimes devastating effect.
And let's start with a number that really grounds us in reality: 31%. That's the 5-year survival rate for ovarian cancer when it's caught at a late stage. It's a sobering figure, and it's exactly why digging into the root causes is so important.
So that leads to a huge question. What if that risk, that ticking clock, wasn't just random chance? What if it was actually written into the blueprint of our cells from the moment we were born? This idea takes us from the big picture of a disease down to the microscopic world of a single gene.
Now, to understand what goes wrong, you first have to understand what's supposed to go right. And for that, we need to meet the hero of our story, an amazing gene called BRCA1. You can think of it as one of your body's most critical defenders. See, BRCA1 is what scientists call a tumor suppressor. Its whole job is to act like a guardian for our genome. It patrols our DNA, constantly looking for damage or breaks, and it coordinates the repairs before a cell can ever go rogue and turn cancerous. It's a really big deal.
And this guardian protein doesn't work empty-handed. It has a special toolkit. These sections you see, the ring domain and the BRCT domains, are its essential tools. They're what allow it to spot DNA damage and then tag other molecules to come in and fix the problem. You take away these tools, and our guardian is completely disarmed.
Okay, so we've met our healthy guardian. Now let's zoom in, way in, to the scene of the crime. We're about to see the exact moment where things go horribly wrong.
And this is what's so wild. The massive increase in cancer risk that we're talking about, it all comes down to this: a tiny mistake, a deletion of just 2 letters out of the 3 billion that make up the human genome.
And here it is. This is the molecular evidence, the smoking gun. What you're looking at is a sequencing graph, and it shows the exact spot, right there. At position 185 of the BRCA1 gene, 2 genetic letters, an adenine and a guanine, have just vanished. This specific error has a name: C.185del.AG.
So 2 letters are missing. Big deal, right? Well, actually, yeah, it's a huge deal. That tiny little deletion causes a complete and total breakdown in the cell's protein-making factory.
Okay, to really get why this is so catastrophic, let's use a simple analogy. Your genetic code is read by the cell in 3-letter words, right? Like a simple sentence: V fat cat sat. Now imagine you delete just 2 letters from the word fat. Suddenly the whole reading frame shifts over. The sentence becomes total gibberish: V FTC at sat. This is called a Frameshift mutation, and it makes the rest of the genetic constructions completely unreadable.
So from that one error, things can go wrong in 2 ways. And spoiler alert, they're both bad. In the first scenario, the cell's internal quality control system spots the gibberish blueprint and just destroys it, shreds it, so no protein gets made at all. In the second scenario, if the faulty blueprint somehow slips past the inspectors, the cell tries to build the protein anyway. But what it ends up with is a stunted, useless version that's missing all its critical tools. Either way, the result is the same. The guardian is gone.
All right, so we've seen the disaster unfolding inside a single cell, but let's zoom back out. How does this translate into a lifelong, inherited risk for a person and even for their family?
Well, the answer comes down to a really foundational concept in cancer genetics called Knudson's 2-hit hypothesis. The idea is pretty straightforward. Remember, we get 2 copies of almost every gene, 1 from each parent. For a tumor suppressor like BRCA1, you need to lose both good copies in a cell for cancer to really take hold.
So for a person with this mutation, hit number 1 is inherited. It's there from birth. They start life with every single cell in their body already down 1 copy. They only have 1 functioning guardian gene, where most people have 2.
And hit number 2, well, that's just a matter of chance. A random mutation, a bit of bad luck over a lifetime, that happens to knock out the 1 remaining good copy in a single cell. And once that happens, that cell has lost its last line of defense. It's now free to grow out of control.
This is incredible. This is actually data from Alfred Knudson's original 1971 paper that proved the theory. That steep curve shows people born with 1 hit. You can see they get cancer much earlier because they only need 1 more random event. The flatter curve is people who started with 2 good copies. They need 2 separate random hits, and as you can see, that just takes a lot longer to happen by chance. It's such an elegant piece of science.
So knowing all of that,
how do we actually find this tiny 2-letter fault in someone's 3 billion-letter genome today? Well, it takes some pretty sophisticated modern-day detective work.
First up, you need a DNA sample. Blood is kind of the gold standard for this kind of inherited testing, but, it's invasive. Saliva is way easier, non-invasive, but sometimes the amount or quality of DNA you get can be a bit hit or miss. And then you can use tissue from a tumor itself, but that DNA is often pretty damaged, which can make the analysis tricky.
Okay, once they have the DNA, scientists bring out the big guns. They use powerful tools like next-generation sequencing, or NGS. Think of it as a super-fast, super-sensitive spell checker that can read the entire BRCA1 gene, looking for small mistakes, like our 2-letter deletion. Then there are other tools, like MLPA, that are great for finding bigger missing chunks that NGS might sometimes miss.
And at the end of the day, this is why it all matters. It's not just an interesting scientific puzzle. As the researchers themselves put it, understanding the stuff at the molecular level is what drives personalized medicine. It allows for targeted treatments and focused screening for people who have these mutations, and that ultimately improves lives and it saves them.
So we've just followed the incredible story of 2 missing letters, 2 characters out of billions that can completely change a person's life. It really makes you wonder, doesn't it? How many other profound stories like this one are still waiting for us, hidden in the vast, beautiful code of our DNA?